Native Plant Society does. And oh, the lady told me recording is in progress. Okay, so and I'm excited to see that a couple of the students that generated the data that I'm going to talk about are actually here tonight for the presentation. So just to jump right in, as Dan mentioned, it's been a minute since I graduated from school. Furman, I'm a faculty member at Furman now, but I've been a faculty member at a couple of other universities. And throughout my career, some of the major themes in my lab have been conservation, first and foremost, reproductive ecology, as the clone theme might suggest, and biogeography, why plants are where they are. And so the work that we do in our lab almost always has some form of field component, so field work. And this is from the Cedar Glades in Middle Tennessee, where we were doing some demographic work with some folks from the Missouri Botanical Garden, actually, on a federally endangered species called Pine's ground plum. And then we also almost always have a lab component. So almost everything I do has a genetic basis. And so we're combining ecology and genetics to try to address ecological and evolutionary questions, and primarily in plants, although I have worked in some other organisms in the past. So right now in our lab, we're actively collecting data on 3 projects. The first one is a federally endangered legume from those glade sites that I mentioned, Dahlia foliosa, the leafy prairie clover. That one's not clonal, so it doesn't really fall into the category of the stuff I'm talking about tonight, but it is a fascinating study in biogeography and reintroduction efforts. But the 2 stories I'm going to be talking about tonight are bunched arrowhead and Oconee bells, which I'm sure many of you know quite a lot about already. So we study clonal plants. A lot of what we do is about clonal plants. So what exactly does that mean? I'm going to give you a little bit of background on that before we jump into the stories of the plants themselves. So there are a number of different terms that are often applied to clonality. You'll sometimes hear vegetative reproduction or vegetative growth, clonal reproduction. These terms are all used interchangeably, but they have slightly different meanings. So some folks suggest that you shouldn't use any terminology with the word reproduction in it, because technically we're not talking about reproduction in the sense that we think about sexual reproduction. And so what's probably more accurate is clonal growth or vegetative propagation. But we use all these terms interchangeably to talk more broadly about the impacts of this strategy in plant systems. So what are we talking about? We're actually talking about the vegetative production of genetically identical individuals, but then those individuals can, with time, become physiologically independent and have the ability to reproduce on their own. Okay, so we'll look at some examples of this in just a second. So why should we care about, I'm sorry, there's a gnat in my office. Why should we care about clonal reproduction in plants? It's actually quite important. As it turns out, most plants, as any of you that do any gardening may already know, most plants can reproduce both sexually and asexually, okay? Something like 80% of all plants, and perennials in particular tend to have these mixed strategies like this. The most common mechanism for asexual reproduction is clonal growth. So most species can actually do some form of vegetative propagation. Interestingly, a lot of data suggests that clonality is really important in species like aquatic species, which again is fitting with the stories that we're talking about tonight. But in general, clonality is thought to be really important in situations where maybe they're limited mating partners for some reason, there's not enough individuals, that can mate based on their genetic background or some number of other issues. But particularly in situations where the environment is not favorable for seed set or germination or seedling establishment, which is sometimes the case in an aquatic environment, right? Or in other extreme environments, clonality tends to be quite prevalent as a consequence of these. Basically, if you can't successfully reproduce with sex, you got to have some way to hang around or spread out, right? And so that's kind of the strategy that we see here with clonal growth or clonal reproduction. So when we talk about clonality, we have to think in terms of defining an individual, okay? If we think about conservation, a lot of what we do in conservation is dependent on how many individuals there are in a population and how long do those individuals persist in those populations. That's a really important component of a lot of recovery plans. Well, when you're dealing with clonality, you have to redefine what it means to be an individual. So we're going to take a look at an example from strawberry, which most everybody's familiar with. In strawberry, we have this nice figure here. And I'm just going to quickly go through the basic structures that are labeled on the figure, just so that everybody's on the same page. And so here we have the basic structures of a shoot, or what we think of as an individual when we're looking at plants in the field, right? And then here we have a stolon. So a stolon is an above ground stem that connects different shoots, okay? So how does that all relate to clonality? Well, the individual shoots we refer to as ramets. And ramets can potentially be physiologically independent, but they're initially born from that connection through the stolon, okay, in the case of the strawberry. The stolon can disappear over time, but each ramet has its own root system. So it has the potential to be independent. It has the potential to reproduce. But ramets of the same unit are genetically identical, okay? Because they're clones, they're clone mates. So we call that clone a genet. So a genet is all the ramets that were derived from the same sort of mother plant, if you want to think of it that way. Okay, so when we talk about individuals in a clonal setting, if we're looking on the landscape and counting individual plants, what we're really counting is ramets, because we can't see how many genetic individuals are present simply by looking at a population of plants, okay? This will be important in when we're talking details of the systems in just a little bit. Okay, so there are also different strategies for clonal growth. So there's some species that's what's called a phalanx strategy, which means basically the ramets of a clone or the ramets of a genet are all tightly packed around their parent plant. So they're not spreading out very much, which means in terms of the landscape and what that means for genetic structure in the population, you're going to have pockets of the same genetic individual, and they're not going to be interspersed with other genetic individuals. Okay, so that would be a phalanx strategy. Alternatively, you could have what's called a guerrilla strategy, and that's where the clone will just basically intertwine with other clones in a population, okay? And so what that means from a genetic perspective is that you have an intermingling of genotypes in space in that population. Why am I showing you this? Because in most species, we don't actually know what their strategy is. And so when we go in the field to try to sample for genetic work, it can be very difficult to determine an appropriate strategy so that we can understand, get a good picture of what the genetic structure in that site looks like. And you can also imagine from an applied perspective that these two different strategies can lead to very different ecological dynamics in a population of plants, which then has implications for conservation management and things like that. Okay, so what dictates those different strategies? We have what are called organs of propagation. And these are going to be all the different ways in which you might see this clonal growth. Things like tillers and corms and bulbs and rhizomes and tubers and root suckers and stolons. And so all these different structures that plants have that can allow them to vegetatively propagate. So what this figure is showing you is the spectrum of strategy from phalanx to gorilla and how it relates to those different types of organs that plants can have. And so most of what I've studied in my lab over the years has involved species that have either rhizomes or root suckers. I worked on American beech trees for a long time, which actually can clonally reproduce through root suckers, kind of like the aspen out west does. And then other species with stolons and runners, like we mentioned with the strawberry just a minute ago. So all these structures are developmentally different, but they all result in vegetative propagation, and the extent to which their clonal individuals spread out is dependent on these structures, which structures they actually have. Okay, so that's all just kind of the basics about how we can think about how clonality works. I can think of lots of great reasons to be clonal, right? We talked about if you're in an environment, in a difficult environment, maybe you want to be able to reproduce clonally. But how might clonal reproduction be a disadvantage? Let's look at a couple of ideas with that. First of all, it's been pretty well documented that there has to be an energy trade-off between sexual reproduction and vegetative growth. If you want to produce flowers and fruits, that actually requires quite a lot of energy. And so if you're investing in sex, basically, you can't simultaneously be investing in an equal amount of vegetative reproduction, okay? Alternatively, if you're rapidly expanding through vegetative growth, you can't then give as much energy to flowers and fruits. And so there is a trade-off there between those two sort of general strategies. Other data have indicated that clonality can potentially cause problems with pollination and mating. So there are some systems where for individuals of the same species to be compatible in terms of a mating situation, they have to have different genotypes. And there are certain genotypes that cannot mate with one another and successfully result in seed. So if you have a lot of individuals of the same genotype as a result of clonality, then that can obviously negatively impact, in some situations, successful pollination, successful mating, and then subsequently successful production of offspring. And for similar reasons, then you might see reductions in fertility, which is going to also negatively impact sexual reproduction. Ultimately, we also see lower genetic variation in clonal populations because you're generating lots of genetically identical individuals. And ultimately, if we're talking about rare species, genetic variation is important, right? We know that that's a key to long-term success and evolutionary potential for those organisms. So we really have to stop and think about, okay, well, what role then is clonality going to play in a system like a rare plant, and what does that mean for how we treat those species? So the questions that we're asking right now are, one, how important is clonality in the population structure of rare plants? And then two, what implications does this have for species recovery and conservation in those systems? Okay. And so the two systems that we're going to be addressing specifically tonight are the bunched arrowhead, which is a federally endangered species here in the upstate of South Carolina and a little bit of western North Carolina. And the Oconee bells, which I've listed here simply as Shortia spa, because you might be aware that there's more than one species here. Just in the last few years, varieties of the species have been recognized as two distinct species, and I'll talk about that. So I'm going to spend a little bit more time on Oconee bells than I will on bunched arrowhead, simply because it turns out the Oconee bell system is actually much more complicated than people may realize. And so there's some different angles to the story that I want to kind of hit on tonight. Okay, so in both cases, we're using the same methodological approach. So I'm just going to give you sort of an idea of what our workflow looks like, so you can understand maybe what it is that we're actually doing. So we start out by scouting out a population. So here are two students from my lab group. Well, can y'all tell what they're looking at? They're looking at Oconee bells, and this is in Devil's Fork State Park. And so we're actually in the early stages of assessing, how big is this population and how is that going to influence how we might sample for genetic work, so that we can try to capture the best overall idea of what that population looks like from a genetic perspective. And I will tell you, with Oconee bells, it is a nightmare to assess extent of population, because if you've ever been in those sites, it just goes forever. It's like a continuous carpet of plants. So once we have determined what we're going to consider our extent of population, then our goal is to sample a minimum of 24 individuals per population. And the reason for that is a technical one that I won't go into, but just know that that's our target number. If it's a really big population, depending on where else we're planning to sample, we might do 2 rounds of this. And we generally try to take a transect-based approach, where every individual plant we're sampling, individual ramet, is 1 meter away from the next one that we're sampling. Okay, and I'm going to show you a map of this in just a second, but we're trying to maintain that minimum 1 meter distance because we just really have no idea at what scale clonality plays a role here. Are clones really small? Are they really big? We have no idea. And so we're trying to overlay this sort of multi-layered scale so that we can get an idea about that. So the picture up top there is are a couple of students working in a bunched arrowhead population. You can see the orange flags where we've flagged off where we're going to sample individual ramets. And then the population below there, that image below, is actually students sampling Shortia brevistyla, which is endemic to a single county in North Carolina. You can see the habitats are quite different. The bunched arrowhead sites are flat. They're super mucky, which can be a challenge, but they're really flat, right? But then with Shortia, we have, they're more pleasant from the mud perspective, but they can be pretty challenging sometimes in terms of topographic variation, particularly for brevistyla, less so for Shortia galacifolia. Okay, so what does sampling look like? This is an actual map that we drew in the field to reflect the individual plants that we were sampling. And so you can see that we have literally a minimum of 1 meter distant between individual plants. We make notes if there's no plant at that point on the transect, and we try to get as close as we can to that. So the idea here is that by mapping everything physically, we'll be able to go back and say, okay, this sample at this particular spot has the same genotype at this sample at this other spot, and we'll be able to actually estimate how much distance there is between clone mates of the same clone. And then in terms of the genetic work, what are we using? We need a single leaf from each individual ramet that we're sampling. And you can see Skylar there, who's all thumbs up and happy to be sampling in the field, but she has a tiny little silica packet like you might have in a new bag that you buy. You'll have those little silica packets to keep things dry. That's what we use to rapidly dry leaf material so that it's preserving the DNA in the leaf. And so we're not taking a whole plant. we're not doing anything really to harm the plants. We're just taking a small piece of leaf material, and that'll be what we use for our genetic work. Okay. Just moving y'all around so I can see. So then we take all that material back to the lab, and here are a couple of our lab mates in our lab here at Furman doing DNA extractions and amplification of genetic markers. Now, we use a tool called microsatellite loci. I'm just going to refer to them as genetic markers for the duration of the talk tonight. The data that we generate looks something like this. If you're interested in learning more about what this actually means, I'm happy to talk to you about that. But I figure y'all are probably more interested in the plants themselves and what the data mean for the plants you can see on the ground. And so what we can do with these types of data is to actually define genetic relationships between individuals that we sample, so that we don't have to physically dig up a bunch of plants to know who's actually connected to whom, right? And so here I am showing, let's see, this would be Shortia galacifolia, because this was at Devil's Fork State Park. And so you can see the connection there that I literally dug up out of the soil. And you can see the individual roots on those different ramets. So those individual ramets should be genetically identical. But because we have this genetic technique, we would be able to tell you that just based on taking a leaf from an individual sample, rather than having to dig everything up to know how things are related. So here's our team, a really fantastic group of students that I've been working with this year. 2 of these students, I think, are on the call tonight. I believe I saw Lauren and Skylar log in. So y'all say hey to Lauren and Skylar. They've done a lot of work, a lot of good work. But all of these folks have been really excellent researchers and have worked well together, which has been really fun. We've had a lot of good times in the field this summer. All right, so let's talk about some plants. So I'll start with bunched arrowhead. Again, as I mentioned, I'm just going to briefly touch on what we're seeing in this system so that I can spend a little bit more time on Oconee bells, given the complexity that we have going on there. And I'll just say in both cases, the data that I'm presenting tonight are very preliminary. So we developed all of those genetic markers ourselves, which that in and of itself takes time. And then you have to test those markers to see what's going to work for the plants that you have. And not only are things going to work, but can you clearly interpret the information that you're getting from those tools? So a lot of what the students did this summer, we spent all of June in the field, literally all of June in the field. If we weren't in, well, actually Skylar and I started the beginning of June in Tennessee with Dahlia foliosa in the field. And then we came back and alternated between bunched arrowhead and Oconee bells for all of June. A lot of hard work, and then did a little bit of Oconee bells work in July. Most of July and August was lab work. So that gives you a sense of what our summer was like. So with bunched arrowhead then, let's see, what are we talking about here? Oh, and unless otherwise noted, I took all of the photos. There are a few cases where I use someone else's photo and I make note of that. You'll see for Shortia, I don't have any flower photos of Shortia, unfortunately. So I borrowed someone else's. So Sagittaria fasciculata has a super narrow distribution. We know it's a small herbaceous plant. It's an aquatic species found in slow seeps and deciduous forests. So if anyone has ever seen bunched arrowhead in the field, it's kind of funny because most people don't find it that spectacular. But I was so excited the first time I saw it in flower. I thought it was absolutely beautiful. And I emailed a colleague and he was like, well, they're not really much to look at. But but honestly, I thought they were. So they're quite small if you're not familiar with them. And as I said, they're in these seeps. They're not in flowing streams necessarily. If the water's moving too fast, you're not going to find them there. But again, if it's too slow, then you're probably not going to find them there either. So they have a fairly specific habitat. They're endemic to Greenville County, South Carolina, and then 2 counties in North Carolina. So we're talking really narrow distribution. We know that they can reproduce sexually by seed, but they also reproduce asexually by rhizomes. Now, we don't really know how extensive clonal reproduction is in this species, and there are a lot of reports in the literature of people moving material from place to place intentionally as a mechanism to help the species, but there's not always clear documentation of the source of that material relative to where it's going, and we don't know the genetics behind the populations to be able to inform those movements. And so that's one of the things that's a little bit of a challenge when you step into a system where things have been moved around, and you'll see in a little bit, I'll ask for your assistance in that regard. If you know things, if you have anecdotes about, oh, well, so-and-so collected some of this over here and moved it over there. That kind of information is often not published, but can be extremely helpful to know when you're studying these systems. Okay, so what have we learned so far in this system? So the picture that you see there is from our population here at Furman, which is a relatively small population. The transect you see there is running almost the full length, and the population sort of circular. I forget the full length that we were looking at there, but what we're seeing is, first of all, we've finished that initial screening where we selected our genetic markers. I talked about that a few minutes ago. And we selected so far 7 genetic markers. We'll be continuing to add to that number. The more markers you have, the more resolution you have in the data set, and the more confident you can be in your conclusions about what's a clone and what's not a clone. And so what you'll see here is in red, I'll present our sort of take home points, our findings. So one thing I did is asked Keith Bradley if he could grab me a close relative of Sagittaria fasciculata. And we happen to have one here on the coast called Sagittaria weatherbiana. And so Keith grabbed some material from a couple of individuals of that, and we use that as a comparison to our sampled bunched arrowhead locally, because the expectation is they should be genetically different. And if they were not, then our markers are probably not an appropriate tool for what we're trying to do. So the good news is that weatherbiana was definitely distinct from our target species. So that gives us some confidence. And it means that we can potentially use some of these markers to look at broader questions of relationships in closely related Sagittarias, of which there are quite a few rare ones that are closely related to our local bunched arrowhead. So some interesting questions there. We are seeing genetic variation within a site and between sites, particularly at the Bunched Arrowhead State Heritage Preserve, at the Blackwell State Heritage Preserve, and the Bellevue Springs Preserve. We're seeing variation in all of those sites, and that's based on the small data set that we're working with in this preliminary screening. Interestingly, this Furman site that you see in the photo, based on our very preliminary data, I can't say that enough, could represent a single clone. So the data that we've collected so far suggests that there is little or no variation at all in that population, which would suggest that that population represents a single large clone. Now, again, we've got more work to do, but what we're seeing is that over a distance of more than 10 meters, the individuals that were included in the initial screening are genetically identical. So that has some considerable implications for conservation. If your whole site is just a single individual, what does that mean about long-term success of that site? So that's where we are right now on bunched arrowhead. More to come on that story as we continue collecting additional data. Now, I want to spend this last bit of time talking about Oconee bells. As I mentioned, there are 2 species, and we are actually working on both of those species. So first of all, if you know Oconee bells, you probably are looking at this map and going, what the heck is going on? Because that is not accurate. So this map comes from a website called BONAP that some of you might be familiar with. And from time to time, there's some odd reporting in there. But the intent is to show you county level occurrence data. That sort of lighter green or aqua green color in Tennessee indicates, I believe that is a native species that has been naturalized there. So meaning, even though Shortia is native to the region, it is not native to those counties, but perhaps it's been documented there. So those would be examples of known plantings that have been established. All right, so again, small herbaceous plant, 2 recognized species. These were previously 2 varieties of the same species. And in 2019, Chick Gaddy and his team used molecular data in combination with morphological data to identify these, raise them as 2 separate species. So we have Shortia galacifolia, that's the more broadly distributed Oconee bells. That's what you see in Devil's Fork State Park. It's now commonly Southern Oconee bells, okay? But then, and you can see the few counties that it occurs in. The ones with an asterisk are counties with noted occurrences that are suspected to not be natural occurrences, okay? So an interesting thing about Oconee bells is there's been a lot of movement, a lot of people-mediated movement of Oconee bells, which makes things really challenging to sort out. The second species we have here that is particularly of conservation concern is Shortia brevistyla. This is the northern Oconee bell. It is only known from McDowell County, North Carolina. That's the only place in the whole world, okay? So as I said, we are including both of those in our work. They are both considered state listed species, but they don't have any federal protection. And my hope is that through this work, we can provide some additional data that might lend itself towards considering the status of these 2 species and whether or not they should have additional protections. So the 2 species are in fact morphologically different. They are molecularly different based on the work that Chick Gaddy did, and they are geographically separated. So their ranges do not overlap. I believe the known occurrences are about 100 kilometers apart, if I remember correctly. So there is a gap there, geographically speaking. Threats to the species, the biggest threat has already happened, I guess, and that was the formation of Lake Jocassee, which basically inundated more than, I believe I've read as much as 60% of the natural habitat of the species. So at that point in time, some of the quotes I've seen in some different articles just talk about Oconee bells as far as the eye can see and how they'll never be extirpated because there's so much of it. And here we are talking about rarity in the species. There also have some popularity in the horticultural trade, at least in niche markets. And that's something I'd like to explore more. I'd like to know more about who is selling them. And if they're willing to talk to me, what their source material was and how much they sell, all these types of things, and potentially let us genotype their material so we can better understand how it all fits into the broader scheme of things. We've done some work like this in some other rare species. So reproduction is complicated in this system. These plants do reproduce sexually by seed, but they reportedly have a mixed mating system in that they can self-pollinate or they can outcross, meaning they can pollinate a different plant. And so, of course, this is going to have impacts on the genetics of the system. They're also apparently pollen limited, which can reduce reproductive success. And we know they can reproduce clonally. I've seen it reported as stolons or rhizomes, depending on the paper. So if you're in the field with the plants, you can see why the confusion on that. We don't know how important clonality is. My assumption after walking into Devil's Fork was, holy cow, attack of the clones, right? It's gotta be one big clone. It's crazy. It's like a big carpet. I've spent so much time with these plants this summer that now when I see English ivy here in town, I'm thinking I'm looking at Oconee bells. I mean, that's what we're talking about here is a thick carpet of this stuff. So what do we know? We've done that initial screening. We've included individuals of brevistyla from 3 different geographic locations within the county. We've included 3 locations of galacifolia, all within Devil's Fork, so relatively small geographic space. We've got 9 markers so far for this. And what are we seeing? First of all, the 2 species are definitely genetically distinct. Our data strongly support what Chip Gaddy and his team said in the 2019 paper. In fact, 2 of the 9 markers that we've developed don't even work in brevistyla. This is not a fluke. It's not a technical issue. It's an evolutionary difference between the species, okay? And 4 of the remaining 7 markers indicate that there's no shared genetic information between the 2 species. And again, this is based on our small subset of data that we're working with right now. What else have we seen? Brevistyla, again, that one county endemic has extremely limited genetic variation, potentially no variation. So with the exception of one minor piece of information that we've collected so far, we've detected literally no genetic variation among individuals of brevistyla across 3 geographically separated sites. So that's pretty shocking, actually. And at this point, I don't know how to interpret that. I don't know that it could all be one clone. It seems like there should be some other explanation there, but we need more data before we can say anything for sure. What I will say is Gaddy's work using different types of genetic tools did find very limited genetic variation in brevistyla. So this is consistent with previous findings, which means we're not just totally off the rails here with what we're seeing, but I'm looking forward to expanding our data set so we can get a better sense of what's going on there. Galacifolia, the more widespread one, has quite a bit of genetic variation, which actually was kind of a surprise. Remember my assumption of attack of the clones? What I'm seeing in this preliminary data set is that we do have genetic variation within the sites and among the sites. There is some indication that there's some clonality there, but it's not the predominant thing that we're seeing, at least based on this tiny little data set. Okay, so what does this all mean for conservation across these 2 different projects? First, it does look like clonality is going to play an important and complex role in these 2 different systems. And so we really need to think about what an individual is for bunched arrowhead or what an individual is for Oconee bells in terms of how we deal with recovery and conservation. In particular, for me personally, I feel like the lack of variation that we're seeing in that narrowly endemic brevistyla, Shortia brevistyla, is a real concern, and that we really need to be advocating for more protections there. But again, we don't yet know why it looks like that. We need more data to be able to say something. And so we really need to expand the scope of our geographic sampling and the scope of our molecular sampling. We have a lot more samples, a lot more data that the students are actually collecting now. But I want to expand it even beyond that to get a much better resolution of the whole system in both cases. So how can you all help with this work? There are actually quite a few ways. One, I mentioned earlier, the oral histories on localities. So if you see something, say something, right? That's a saying. If you know something about a particular locality, or you know something about individuals who have moved material between localities in either of these systems, that information can be super helpful. And again, it's not the kind of thing that's always recorded in publications. If you have localities of these species on your private property or know people who do, and you're willing to work with us on that, it would be excellent if we could get access to those sites to include them. Assistance with field work. I've been talking to Kay Wade about getting on her boat and riding around Jocassee so that we can collect Oconee bells on the edge of the lake there. And so things like that can be hugely helpful for us, right? That's not something I would normally have access to. Also, just being in the field and helping us collect leaf material can be helpful. And of course, financial support for cost of research is huge. The genetic work is quite expensive. I should have calculated how many dollars are sitting on that lab bench, but I didn't think to do that. It's a lot of dollars, I'll tell you that. So these are all different ways that folks can contribute in terms of the financials. I'm pursuing grant money so that we can continue to expand these projects. But if you know somebody that just has a bunch of money sitting around and they just don't have any idea what to do with it, they could always contribute to this work. And with that, there are a lot of folks that I have to thank because we've had a lot of assistance with the development of the project, with the field work, and then the students in the lab, of course. And so I just, with that, I'll stop and I'll be happy to answer any questions that anyone may have.
Hi, Ashley. I was wondering, how do we get in touch with you if we have a population? Before we started, I shared that I have about a half an acre's worth of Oconee bells growing on my property up off 130 north of Highway 11.
Okay, I'm putting my email address in the chat, and I would just ask you to reach out to me directly by email. That's great. That's the kind of stuff I like to hear about.
Okay, I'll shoot you an email.
Thank you. Other questions?
Yeah, what is the legality of selling plants of Oconee bells?
Yeah, so I don't actually know the answer to that. That is something I need to dig into further. And I did see on one website that it claimed that it's illegal to sell Oconee bells unless you're a permitted seller. I don't know if that's accurate or not because it's not a federally listed species. So I honestly don't know. That's something that I would like to know more about. I can tell you this species that I had worked on previously that is horticulturally important is Florida anise, Alyssium parviflorum. It's endemic to a handful of counties in Central Florida. And you can buy it at Lowe's. You can buy it at our Lowe's here in North Greenville. And for me to sample it in the field, though, it was a state listed species. I had to have multiple permits, landowner permit, state permit, etc. But you could go buy it at Lowe's and it was planted all over the place. And so in that case, I don't think there was any regulation at all of that species. I do not know if the same thing applies in this case. I've heard numerous stories about how when Duke Energy was planning to impound Lake Jocassee, that people were invited to come in and collect the plants from the valley and take them out of there to save them, as it were. And so there are some reports that some of these stray populations around the edges of the known range could actually be a consequence of those early collections out of the valley. So that's the kind of stuff that, you know, sort of word of mouth or myths or whatever you want to call them, that the more of that kind of information that I have, the more helpful it will be. I've made some connection in other states with folks who are aware of errant plantings, like that Alabama population is in a state park and they think it was planted, but they don't really know for sure, and it's naturalized there now. So I've got some material coming from that site, for example. So yeah, it's a really fascinating story between the conservation angle, the lake impoundment, and then the movement of plants by people. It's just a really interesting system, I think.
Actually, I assume you're aware of Joseph Townsend's work with.
I have heard. I never got a chance to meet him, but I am aware that he did. Am I correct in thinking he was able to propagate them by seed?
Yes.
Okay.
A lot. I think a lot of what the plant material that he had is now in the hands of Ryan Merrick, and Ryan would probably know some of the provenance of those things.
Okay.
He and
And where is
Ryan Merrick?
Where is Ryan?
M-E-R-C-K, Ryan, M-E-R-C-K, Merck. I can send you his email address.
That would be great. Thank you.
I know of a planted population at Pearson's Falls, and the story that I heard was that it was during the time that the lake would be flooded, and that the garden club was asked to come and help save some plants. And
okay,
it's worked its way downhill. It's multiplied.
Yeah, that's kind of what I'm hearing is when people have planted these things, especially if they were planted a long time ago, they spread, right? That's what we see.
Yes.
And so it'll be interesting to see if these sites where they were known introductions, is it one genotype or has it managed to reproduce sexually over time and you have different genotypes? So yeah, any of those kinds of populations, I'd love to know about them. I know at Devil's Fork, we did get a couple of individual samples that the ranger there told us was supposedly from pre-inundation down in the valley, and it's now planted in a little garden there right next to the visitor center. So we have those individuals, but it would be great to have as many of those types of examples as we can, so that especially when we try to figure out like the Alabama stuff or the Georgia stuff, if it matches something from that basin, then we have a strong chance of being able to say, well, those aren't natural occurrences. They were clearly transplants from there.
Right.
Ashley, could you post your email again?
Yeah, it's in the chat, but I'll it's just ashley.morris@furman.edu.
Okay. Is the Dixie, is the South Carolina Botanic Garden population a plant rescue from the Gorges area?
Yeah, it sure is. Yeah. Have you gone to talk to them?
I have not. I will make a note that I need to do that. Dixie, who would be my contact there?
Maybe Kathy Bridges.
Okay. Thank you.
Uh-huh.
And this is Rick here. Up in the East Oconee Valley, I know where there's several large mats in the East Oconee Valley.
Okay.
Up on near Cane Creek.
Will you shoot me an email so we can start a conversation about those?
I sure will. Yeah.
Thank you.
Yeah.
Yeah. See, this is great. This is exactly what I was hoping. This kind of
We're out
having
there.
these kind of connections is awesome.
Yeah.
And if you'd be willing, I'll take you on a field trip to see some pink bells.
I
Sometime.
would love for you to take me to see those pink bells. I've seen some photos. I would love to see them in person. I tried I've only been in South Carolina for 2 years. Oconee bells were new to me in South Carolina. But when I learned they were clonal, I said, well, obviously that's right up my alley. They're clonal and they're rare. And so last year, I tried to catch them in flower, and like the narrow window of time happened to be when I had to travel. And so I got to see like one flower that was open. And then while I was gone, everything else opened. And by the time I got back, it was done. So I definitely, that'll be my goal in the spring, is to go out and see this for myself.
When you take a sample of for genetic study, is like a hole punch, or what size of a sample of a leaf do you do?
So for the bells, we typically take one whole leaf and we just tear it into shreds and put it in the silica desiccant. And the reason we do that, the amount of material that you need for an actual DNA extraction really is more like a hole punch. I always tell the students, use about as much as your thumbnail. But let's say something goes wrong with that extraction. We put all that time and energy out in the field. If we only got enough for one extraction, then it's totally wasted if it goes, if it goes south. So we have that material in the lab that we can always go back to if we run out of DNA or if there's a problem with the DNA. But we only pick, a small leaf. For bunched arrowhead, those leaves are quite large. So depending on the size of the leaf, we may not even take a whole leaf. And they're a little bit different in terms of their thickness and the nature of the leaf. So they dry differently for the two species. It's so you have to you don't want to put too much leaf material in there. It can actually negatively affect your process.
So I would like to offer a theory about the ones at Pearson's Fall. They are in a narrow band that looks like they washed the seed down the hill. So it probably is worth looking at.
Yeah, that sounds good. I've heard similar things around, I've heard there's some Caesar's Head, Table Rock area. And that the suspicion is they were planted in some of those I guess there's some cabins or homes up there or something, and perhaps they're escaped from that. And what I've been told is it's like they're sort of oozing down slope, like you're describing.
Right. Well, they're very happy.
Well, actually, these are some very cool plants, and it's a lot of good information that we've learned about them today. And we really appreciate you taking time and telling us more about the fascinating world of nature out there.
Well, thank you all for having me. I appreciate it. I've enjoyed talking to you, and I look forward to hearing from you with your own populations and ideas moving forward.
Okay, if you want to see that address on the chat box, put your cursor down at the bottom of the screen and you'll see all these things pop up, and click on chat and you can see her address there on the chat box.
So for me, who's working from an iPad, would you repeat it one more time?
Sure. It's ashley.morris@furman.edu.
Okay, got it.
And if it's .edu, you can count on it.
Well, I don't know about that.
Thank you, Ashley. Thank you,
Thank you,
everybody, for
Dan.
joining us.
Thank you.